Hey guys, this is Mr. Chemist from Wayzata High School, just talking to you about patterns of inheritance. We're in chapter 9 right now at the beginning of the chapter, and this is the lecture you should find about monohybrid crosses. It's a little bit long, but once you get the basics down, you should be able to do quite a few genetics problems, use the vocabulary correctly, and know some of the history of genetics here. So I'm going to go to present here so you can see what we're working on. All right, here we go. So you should be able to see my screen here. Here we are in Canvas with the BioX page. Yours should be set up like this or similar. And then you want to click on this funny looking Austrian monk. He's the father of genetics, Gregor Mendel. And that should take you to the modules for genetics here. You can see the unit plan here that will link you to the schedule, which will tell you what's going on this week. And then the genetics resources, if you want to pull up any of the PowerPoints, homework problems, answer keys, and resources for studying for the test, that's going to be there for you. So I already clicked on the unit plan. Here's the plan for the week. We have monohybrid crosses and dihybrid cross today. We're assuming that you've already read the first bits of the chapter. Objectives 1 through 3 talk about the monohybrids, and 4 through 6 talk about the dihybrids. So we're assuming you read that before you watch this. If you haven't, go back, stop, go back, do that quick, and then come watch my recording. And then the homework tonight is the homework number 1. The first 3 pages of that are monohybrid crosses. You can see that here. And then the last page will be dihybrid cross. So the monohybrid cross should be a review for you. Make sure you get that down. And then the dihybrid cross will be a little bit more 10th grade level for you. And then keep going with the objectives. Just like always, all our objectives are down here at the bottom. So you can see those. And then the vocab is pretty tough. This chapter, there's a lot of vocab words, so really focus on those. But quite a few of them come in pairs, like heterozygous and homozygous. If you know one of those, you can know the other one. Genotype and phenotype. If you know one of them, you can know the other. So hopefully that can help a little bit. There's also word roots down here, quite a bit of Latin and Greek roots here. So take a look at those. Those help people learn the vocab. All right, well, I'm going to go to the lecture here. Let's see. Let's go to this, and I downloaded the 01 Fuller Mendel notes here. So let's go to that one. All right, so chapter 9, you should see chapter 9, and you can follow along on with me, or you can pull it up separately, or you can go back and check it anytime. All right, so the chapter starts out with the patterns of inheritance. We have genetics being the scientific study of heredity, and heredity is how things are inherited from your parents, different traits. And it starts off with the idea of the dogs, and we can breed dogs, and we can figure out what traits we want to keep, and what their parents have probably going to get passed on to their children, and so we can start to breed the types of dogs we want. Here you have the sled dogs. We're going to want some that's fast. We're going to have ones that have well-insulated ears. They're good runners. They're strong. The problem is when we start to isolate for these genes, some other undesirable genes might come along. The one your book talks about called PRA, progressive retinal atrophy. This is a common type of blindness in Siberian Huskies and several other breeds. And some of the things that we learned about breeding dogs, we can then apply it to humans, which is pretty cool. And so the book will come back to throughout the chapter and look at dogs as an example of genetics. All right, so let's go back to the history. Page 146, you have Gregor Mendel. And if you keep your book open too, you can kind of follow along at home, which is kind of fun. Gregor Mendel is the father of genetics, and he's pretty amazing. If they would have had the Nobel Prize back then, he would have got it because he was a genius. All he did was grow peas in his garden and do math. and he figured out what a gene was. They didn't know what DNA was. They hadn't even worked out evolution very well yet. And he figured out how genes are transmitted from parents to children and how they go through several different generations. And then he could figure out what traits were dominant and recessive. And he also figured out that genes segregate independently. How do you do that? Just by growing in peas, looking at their traits and figuring out how they work together. He was a pretty amazing guy. So he lived in Austria. He was a monk, and he took lots of notes, grew thousands of peas. It was pretty cool. Why did they pick peas? Well, they're easy to grow. They have lots of simple traits that are dominant and recessive. And then he could also control which ones bred together. So if he had a purple flower and a white flower, he wanted to see which one was dominant, he could breed those together, and he can control which ones were there. Peas can also self-fertilize, so you can find out what genes they have and do a test cross that way as well. So let's take a look at the parts of the flower here. The petal is going to attract the pollinator, and then you have the male part is the stamen, and at the top of that you would have the anther, which holds the pollen. And then here's the female part of the flower in the center, the light green part. There's just one of those, and that's called the carpel. And down here you would have the ovary where there's eggs. And so Mendel could figure out how to manipulate the sperm and the eggs there to figure out, and then he did tons of times, controlled which flowers bred with which. And then from that he could deduce what it was a gene. So here you can see the anther and the stigma is the top of the female part there. All right, so here's how he did it. Basically, you could cut off the part that produces pollen or which holds the sperm, so that that flower was not allowed to reproduce. Okay, you could just artificially remove that. And then he actually used a paintbrush on other flowers. Whoop, there it goes. He used a paintbrush on other flowers to take this plant sperm, this pollen, and spread it onto the flowers that he did want to breed, that he did want to test and figure out their genes. And so in this way, he was able to control which flowers bred, and he could deduce and figure out their genes. Here's a diagram from the book that kind of goes through the different steps there as well. You cross a purple and a white flower. Which ones you want to take the pollen from the white flower and move it onto the purple, and then you can grow peas. So that this flower would then develop into peas, and you could plant those and figure out what color all the first children, or the F1, the filial generation one, their offspring. What color flowers would those have? And if they're all purple, then you know purple is dominant to white. All right, vocab. Make sure you check these out. This is a picky detail from the text. Okay, character is going to be what feature we're talking about, and trait is which variant. So please be careful. The text picks these out. So flower color would be the character, and then the trait, it would be purple or white. And there's a chart in your book that has several of these characters and traits. You should know some of these basic P characters. A gene is any sequence of DNA that codes for a protein, then gives you your trait. And then allele are different forms of the gene, different versions. Usually there's just two forms of the genes. There's a couple traits that we'll study that have three forms of a gene, three different alleles, but most of them are just simple dominant recessive, two forms. And then here you can see different chromosomes below here, where there are different alleles for white flower, allele for purple. And so maybe you could get this chromosome from mom, and you would get this chromosome from dad, and then you put them together and it figures out what your genes are. And then this pea plant, whatever trait is dominant, that's what color flower you're going to have. Okay, how does this all work together? So here's another vocab pair. You have genotype and phenotype. Genotype is the two letters that you have, the different alleles that you have. You got one from mom, one from dad. And then the phenotype is the physical characteristic. What do you look like? And if you're a pea plant, you're going to have yellow seeds or green seeds. You're going to have tall or short stems. You're going to have yellow seeds or green seeds. So those would be your phenotypes. And then your genotypes are going to be the alleles. And what letters do you have for those? So the genotype is what you get. Okay, the environment and development can also play a role in what you look like and how you behave. And then the activities, genes, and their gene products, this is what's going to happen inside of your cells to make you look this way or start to behave that way. Here's the character and trait again. And then here's the main chart. Okay, this is a key diagram for chapter 9. Make sure you know some of these characters. Color, flower position. Axial means on the side. Internal means the flower is going to be at the end of the stem. Seed color, surprisingly here, yellow is actually dominant to green, even though when we think of peas, usually it's probably green. That is a recessive trait. Round is dominant to wrinkled. Okay, and then let's take the pod all by themselves. Inflated or smooth is dominant to constricted, and green pea pod color is dominant to yellow. So that's a little reverse of the actual peas. And then of course we have the common tall is dominant to short or dwarf. So come back to this one, you'll need this one. It's a good diagram. Okay, all right, generations. Make sure you know some about this, okay? The parent plants or the parent dogs or the parent people, whatever you're talking about and tracing their genes, we're gonna call that the P generation. And then the first filial generation is gonna be the first children or offspring. And then typically you would cross 2 of these again to get the second children or the F2 generation. And that's really easy to do in plants, and Mendel did that. Basically, you would do this because the recessive that are hidden in the first children, the F1, would then be revealed in some of the children in the F2. So this is a good way to unveil those hidden recessive alleles. True breeding is also going to mean that doesn't matter how you breed them, they're also always going to show that dominant trait. Okay. And then hybrid of the offspring of 2 different true breeding varieties. They're usually going to show just the dominant allele in the F1, and then they'll show some dominant and some recessives in the F2. We call that hybrid, a mix of 2 different true breeding. So here would be an example of the 2 true breeding ones, and you could test this to see if they're actually true breeding. What if you cross a smooth and a wrinkled? Well, the first generation of pea plants, they would have all smooth seeds. So what is dominant there? Yeah, the smooth is going to be dominant. But what if we cross 2 of those F1 plants? This is a typical cross that Mendel would do. Well, then we're going to get some of the F2 are going to be smooth and some are going to be wrinkled. So that recessive wrinkled trait that was hidden or masked here in the F1 is going to be revealed again. Also note the math here. It's about 3 to 1, okay? And in your Punnett squares, you'll see that pretty easily, the 3 to 1 ratio. But in real life, that's not always perfectly 3 to 1. So be ready to look for that. Look at this, 2.96 to 1. Okay, well, that's about 3 to 1. We don't always get exact perfect ratios in real life. It's another thing that tricks up some of the students. All right, and what if we cross purple and white? Here's another example. Here's the parents, big P, crossed with little p. About half the genes are going to be big P and half are little p. So all of the first generation hybrid plants are going to have purple flowers because purple is dominant to white. Be careful when you use the letter P as well. It's not very clear. Sometimes I use a cursive letter for the lowercase. That helps me remember, oh yeah, that's lowercase. And then I won't make any mistakes. Also note these gametes on here. That's going to be the sperm or egg. You get 1 letter from each parent. So this purple flower, the only gamete it can give is a big P. And this white flower, the only gamete you can make is a little p. In the F1 generation, then, if we breed two of those plants to get F2, half of the gametes are going to be big P and half are little. So let's breed those. What would we get? You can put that in a Punnett square. You can get big P, purple, big P, purple, big P, little p, purple. And then the double recessive here, two little p's, which can be the white flower, comes back. And so a typical ratio for this, the phenotype would be a 3:1, and the genotype is going to be a 1:2:1. Make sure you're able to write those out, genotype and phenotype ratios. I usually put the genotype first. Here it is again. You can see it just in a picture form. Here's Gregor Mendel in his garden. He's looking like he's working really hard there. He grew a lot of peas. All right, so should we go through the monohybrid crosses? Here's Mendel's hypothesis. I'm on page 148 if you're following along at home. There can be multiple forms of each gene. We call these alleles, okay? And that's just a simple big P or little p, big Y or little y. For each trait, just use one letter. Tall and short, you use big T and little t. Or you could use big S or little s. I'm not good. Some teachers are picky about which letters you use, but I am not. It's up to you. Just be consistent for each character that you're following there. All right, number 2. For each inherited character, an organism inherits 2 alleles, one from each parent. Okay, this is the idea that you get half of your genes from mom and half from dad. And the vocabulary goes here, homozygous. Okay, homo means same. So you would have 2 capital letters or 2 lowers. That would be homozygous, dominant, or homozygous recessive. And then there's only one way to be heterozygous. Hetero means different here. So you're going to have 1 capital and 1 lower. Genes follow the principle of dominance. Okay. Most genes that we're studying here, there's a few special cases this doesn't happen, but all these ones that Gregor Mendel did were simple dominant recessive. So this trait in the dominant column is going to hide or mask the recessive trait whenever we have a heterozygous plant. And that's the principle of dominance. And so a recessive allele is going to get hidden or masked. Here's some examples from humans as well. Can you roll your tongue? I cannot. So I know what my genes are. I'm double recessive, little r, little r. How about freckles? Freckles is actually a dominant trait. If you have freckles, you have at least 1 capital F. I do not. Widow's peak. Do you see that little triangle coming down here? If you have a hairline that has a little bit of that, that's called a widow's peak. If your hairline goes straight across, you do not. You have the straight, which is recessive. Earlobes. You look at your ears. Does it hang free down here? I have the free hanging. Or does your earlobe attach right at the bottom and stick to your, it's a simple dominant recessive trait for humans. The cleft chin. That's dominant to no cleft chin. You have that butt chin there. Hitchhiker's thumb. Can you move your thumb back 90 degrees or not? That's going to be hitchhiker's thumb. I think I have that one maybe. And then dimples. Dimples is also dominant to no dimples. So you might see some of these in the word problems and practice problems. Not sure what they are. A simple search to look that up again. All right, law number 4. A sperm or egg carries only 1 allele for each inherited trait character because the 2 members of an allele segregate or separate from each other during the production of gametes. This statement is now known as the law of segregation. Okay, so there's 2 parts to this. You get 1 allele for each character from each parent. You get half your genes from mom and half from dad. And if you extend that out to the chromosomes here, you would get 1 chromosome of that from, like blue would be from dad, and 1 chromosome from mom, which would be red. Okay, and we'll come back to that in a day or two with the meiosis lecture. And then also, these alleles are going to separate from one another during gametes. So usually the alleles, unless they're really close together on the chromosome, we're going to separate independently. Okay, we call that the law of segregation. Here it is in text version again, if you want to read through that again. All right, next idea kind of talks about the location of genes. Okay, a locus is the singular form of the word, a locus, and that is the location for a gene on a chromosome. So we can figure out what chromosome it is on. Okay, humans have 23 pairs of chromosomes, so we can tell you which chromosome it's on. And then we could tell you like where it is on the chromosome. There's a couple different numbers and letters for doing that as well. Okay, so the plural for locus is called loci, which you see here. Loci with a soft C. Your book shows this diagram. Okay, so here's a P chromosome. Chromosomes have, P's have several chromosomes, but here's one of them. So maybe this blue one, this P plant got from the father plant, and this red one they got from the mother. Okay, so they have two alleles. Here's a capital P, capital P. So that's homozygous dominant here. Little a, homozygous recessive for this trait. And then dominant, one dominant allele, one recessive, big B, little b. That's going to be a heterozygous example there. Okay. So these homologous chromosomes carry the same type of information, but they might have different forms of the gene. Skip that one. And then just here's another pair of vocab words if you haven't figured this one out yet. I usually figure out the genes first, the organism's genetic makeup or genotype. And then you can look at that, what's dominant or what's recessive, and figure out the phenotype, their physical trait, what do they look like? So make sure you know that pair of words as well. We use that a lot. Okay, so then monohybrid crosses. You can go through and look at this one. Here's another example. If we look at pea pod, what if we had a green pea pod crossed with a heterozygous yellow? So little y crossed with big Y, little y. That's going to be there for you. Half of the children are going to be yellow. Half of the children would be predicted to be green. Okay. Sometimes the dominant allele is more common, but it's not always the case. So please be careful. Use your Punnett squares and actually go forth and predict those ratios. There's a little joke about Mendel and his peas. It's a good one. All right, now how do you do crosses? We have several problem sets for you to do this week, and then we can check answers. You can do that on your own, or you can do that with your teacher. We'll have several different help sessions for you, but here's the good steps for solving problems. So I know most people are, when you get good at these, you might skip these. Don't go too fast and make a mistake. Go through these steps, make sure you can do them and use them. You can use these for all the different types of genetic problems we have this week. And here's some problems for you to try. So code the problem. Okay, what does the big T mean and what does the little t mean? Or what does the big P mean and what does the little p mean? So actually go write this out. Big T means tall, little t means short. That's what we mean here for step one, code the problem. Then you need to use, it's kind of like a math problem. Go back to the words, go back to the math problem here, write down the genotypes of the parents. Okay, it might tell you a heterozygous, it might tell you homozygous. They might use words like dominant recessive or true breeding, you might see that. But use the vocabulary and figure out the genotypes of parents. This is probably the hardest step, and I think a lot of errors happen on this one. Determine the possible gametes. For monohybrid crosses, this is pretty easy. You just go back and you put the two letters here. There's no additional step. For dihybrid cross will be an additional step, but just put the gametes here. Here's one, a little y. Here's one, little y, big Y. That's a gamete. Little y would be another gamete. And then do the cross. Step 4, make the Punnett square. Fill out what the children would be. So take a big Y and a little y, a big Y and a little y goes in this box. Make sure you always put the capitals first. That's a good way to stay organized. little y goes here, little y goes in this last box. And you'll typically find the double recessives here, which will have the recessive phenotype is in this lower right. That's a pretty common pattern. And then step 5 is figure out the answer. So this could be in a chart. This could be a ratio. This could be percentages. Look at what the teacher or the problem is asking for. Sometimes they'll just ask for phenotype. Sometimes they'll just ask for how many of each color. Sometimes they'll ask for the genes and the phenotype. So pay attention. Figure out a good method for this. There's a couple of other practice quiz questions here for you to look at. So you can flip through those. And then here's a good summary for you too. Could you explain this cross of tall and dwarf plants using all this vocab? So try that one out at home. Talk to your mom or your dad or even your dog. That's fine. Speaking of dogs, let's go to talk about some probability here. Okay, the last little learning of target talks about using probability in the law of independent assortment. That's page 150, 151 here. So probability is pretty important. It's just like flipping a coin. But how do we calculate that? What if we get 3 heads in a row? Are they independent of each other? What's going on there? So alleles of different genes separate independently. So if we look at the coats and the color vision, you might have to look at those genes separately. We could also calculate the possible phenotypes of a black versus chocolate separately, and the normal versus blind vision separately, or we could put together and do a dihybrid cross. You get some bigger ratios there. So here would be a dihybrid cross of crossing these dogs. You have a black coat normal vision, a black coat blind, a chocolate coat normal, and a chocolate coat blind. Okay, and this is a typical offspring ratio here for dihybrid cross. You want to write this down: 9 to 3 to 3 to 1. When both parents are heterozygous for both traits in a dihybrid cross, you're going to find that this typical ratio in the F1. We'll talk more about that in the dihybrids, but here's a simple dihybrid cross for you with the labs. All right, well, what else do you need to do today? So that is the end of the notes for today. We could also look at what else we need to do. So let's go back to, okay, let's go back to the genetics unit planner. Okay. So on the unit planner here, you can see, we also want you to watch the dihybrid cross or flip through that lecture, and then you can start the homework 1. You can probably do pages 1 through 3 now of the homework. That's the monohybrid cross. So you can look at that one. That is linked here in the schedule. It's also in the resources folder if you go to assignments. I want to dig through that. All right, well, that's the end of your lecture. Thanks for watching, guys. Make sure to like, comment, subscribe below, and we'll see you in the next one.